Yes.
Yes, we do.
Awesome. Hello, everyone.
Good.
Hello.
Good afternoon.
Okay, so then let's start because so yes, good evening to all and welcome to this workshop on deep learning and its application to bioimage analysis. So yes, as I was saying, we have quite an extensive program, so I will keep the introduction short. We have 3 sessions, so each about 20 minutes, and we will take maybe 1 or 2 short questions in between sessions, and if we have time at the end, also some more questions. So Daniel Sage is here with us to moderate, so please ask your question in the chat. And so we will start with the first talk from Ignacio Arganda Carreras. He's a Iker Basque research associate at the Department of Computer Science and Artificial Intelligence of the Basque Country University. And he will start with an introduction to the field. So Ignacio, the floor is yours.
Okay, let me see. How can I join? Okay, I think. From tab. Yes. Okay. Present now. You can see full screen.
Full screen.
Yeah.
I don't think this is possible, but we see it. Yes. Yeah.
Oh, okay. Great. Well, thanks for the introduction, Sebastian. As he said, I'm going to talk about deep learning. So for that, we have to go through the basics of machine learning first, artificial neural networks, before jumping into where we are now, the golden era of deep learning. So I'll try to keep it short, although it's quite challenging to do this in 20 minutes. So the first thing I usually tell my students is to look at it from a historical perspective. No? Let's say that in the past decades or even centuries, thanks to technological advances, we have always been able to automatize tasks that require a significant amount of time and especially repetitive work, manual work for humans. And now, not only thanks to technology, but also to the combination with big data, we're able to automatize tasks that are not only mechanical, for that we have mechanical engineering, but also those that require what we could call a certain degree of intelligence. So what kind of tasks I'm talking about? Well, let's say there are some tasks that are easy for humans and difficult for machines, at least until very recently. For example, let's say face recognition, as I'm showing here. For us, for humans, it's quite simple. We wake up every day, we recognize the people in our surrounding, our familiar faces, our friends, acquaintances, etc. But if you think about it, it has plenty of challenges, right? Because the face of a person changes a lot depending on the position, the illumination, the haircut, if I'm wearing glasses or a hat or not, etc. There are other tasks that are actually hard for humans and not so hard for machines, especially those that are related to large amounts of data. Even face recognition, it's not the same to do it for just a few dozens of faces in our surrounding than to do it for the millions of faces that are available, for example, in the Google or Facebook databases. This is unfeasible for us. So what we call machine learning, data mining, pattern recognition are all these techniques that have achieved great results in this direction, making what we call intelligent systems, well, a very important part of not only research, but also business models. You have now these 2 fantastic words, machine learning, in your resume. Well, it's a big plus for it. But it's nothing new. It was already defined at the end of the 50s by Arthur Samuel, who said that is the subfield of computer science that gives computers the ability to learn without being explicitly programmed to do so, right? So what does it mean in practical terms? Well, it means that whichever data we have, let's say we have some data points, I always give the same example to my students. Imagine that you have an experiment where you know that you have 2 types of cells, green cells and red cells. Well, what machine learning provides us is an automatic way of assigning those labels, those classes, those categories to the points to say, okay, these are green and these are red. And to represent them in a space like this, in a graph like this, we usually have to take some measurements out of our samples. In this case, we could think of maybe the width and the height of the cells, or the area, or the intensities, etc. And then we represent them, for example, here in a 2D space like that. These measurements are what we call features. So this is a representation in a feature space. Based on this, we can differentiate mostly two types of machine learning. One that is called supervised learning, where we start with data samples from which we know the original labels. We know which ones are green cells and which ones are red cells. And we have unsupervised learning, where we just have our measurements, our cells, but we don't know the original categories. This is unsupervised learning. But the target is basically the same. Sorry. Oops. Basically, if we have a new point, the idea is in the supervised learning approach, we would like to assign it to either the green category or the red category. In the unsupervised approach, we would like to say it belongs to this group of points, maybe this cluster number 1 or this second cluster of points over here. So summarizing, in supervised learning, we have data with labels, and the target is to build what we call a model or a classifier to automatically label new data. Okay, and we do so by training on these non-labels. If the labels are not discrete, they are not categories, then we usually what we have is a regression problem. For example, imagine that we have images of digits, and we want to recognize to which digit belongs each image. Well, we have only 10 digits from 0 to 9. So there are 10 categories, 10 classes. This is a classification problem. If I have facial images and I want to estimate the age of a person based on the image, well, the age is a real positive number. It could be 15.3, 35.7, or so this is a regression problem. In the supervised learning, as we said, we don't have data, we don't have labels. The data comes only with the measurements, with those features. And the idea is to model the data to discover groups of samples, or we say are clusters. No, that's why we call it a clustering. So a few concepts that we have always in machine learning are a feature vector. No, every sample is represented by those set of measurements that we call features. That's why we call it a feature vector, which can be numerical, but also it can be qualitative. And in supervised learning, we have a set of predefined classes. No, for example, in the digit example, we know that there are 10 categories, no? Only 10 digits, and every sample belongs to only one of those categories. No, a 3 can only be a 3 and not a 3 and a 7. And hopefully, all our samples that belong to the same class, they are similar among each other, and also very different from the others. This is the ideal case. Sometimes, well, we get number ones that look like number 7s, and then that's where the fun starts. So a typical classification problem is what we call a binary classification problem. Imagine that we have images of biopsies, no? And we want our model to tell us if the tissue in the image contains is tumorous or not. Well, we have tumorous and non-tumorous classes, so then we have two classes, binary classification. Then we have the concept of data set, which is just the set of samples or the feature vectors plus the class if we are in the supervised framework, or without the class if we are in the unsupervised framework, right? So far, so easy. So the idea of the classifier is to have a function that relates our feature vectors to the labels. Okay. Actually, you may find in the literature that some of these classifiers they take a decision in 2 steps. Let's say, no, they first have a soft decision. Maybe they give us the probability of the sample belonging to each of the classes, and then the final decision, the hard decision, where they say, okay, this is definitely a 3, no? Or you could say this is 50% 3 and 50% a 7, something like that. So when we say that we train the model, that we train the classifier, what we are doing is adjusting the parameters of the function to minimize the error or the cost function. No? For example, the error between the expected output and the original output. So you can ask, okay, how do we evaluate the performance of a classifier? How do I know that this classifier is good or bad, or how do we know that how it compares with the others? So for that, we do something very simple. It's called a confusion matrix, where we just count. No? We apply our model to our samples from which we know the original labels, and we compare them. So we imagine in this case, we have, let's say, the positive class and the negative class. Imagine that we have this binary classification problem. So we just count how many of the actual positives were predicted as positives, so that are true positives, and otherwise they are false negatives. And then we do the same with the false positives and true negatives. We're actually now very familiar with this terminology with the, for example, the COVID test cases, right? So we just count this. We know the number of total positives and the number of total negatives in the real case, and based on that, we can extract plenty of metrics. No, the simplest ones. Well, we can just measure the error rate. No, how many mistakes I made out of the total number of samples, or the accuracy, which is the opposite. How many hits I made out of the total number of samples. But sometimes you could find in the literature that, some other metrics that are more interesting, especially when you get imbalance in the distribution of the samples in the classes. No? So you may be more interested in the sensitivity or the specificity, etc. Okay, so once we know how to do this, then we have another question. Is okay, now I have my data points and I want to build a classifier. How many of those points should I use to build the classifier? Imagine that we have a set of points like this, and we would like to have a classifier that models this distribution. We could be tempted to use all of them, but this is usually risky because we could fall into a problem of overfitting, where we adjust our model too much to the training data points. So a new point, unseen point that belongs to the distribution, it wouldn't be considered as part of it. Okay, this is a problem that we call overfitting. So to prevent this, there's simple solutions. The typical ones is to partition your data into two subsets, no? One for training and one for testing. You only train your model, you only adjust the parameters of that function in the training set using the training samples. And when it learns, you compare or you evaluate in the test set. Okay, so the model has never seen the samples in the test set, so it's a very good idea, or it will give us a very good idea of how good the classifier perform in unseen data. If this partition is tough to do, or we don't have enough data or very few samples, there are other solutions. For example, a very common alternative is called the k-fold cross-validation, where we subdivide our data set in k subsets. Usually k is 5 or 10. No? And here I put an example with 5. And then we select 4 subsets for training and 1 for testing. And we train one model like this, and then we repeat this 4 more times, training from scratch one model. So we have at the end 5 different performances on the different subsets of tests, but we usually provide the average as the average result. No? And this is to prevent a situation, for example, where we have all the easy samples in the test set or the difficult ones. So if we just partition that way, we would be having a very bad estimate of the performance of the model. A very extreme case is when we have very few samples, and then we do what we call leave one out. No? We just test on one single sample and we train on the rest. Okay? For example, you just have 10 videos, and then you cannot just do the partitioning like this. So take 1 video for testing and 9 for training. So before deep learning, what we usually did was to, okay, imagine that we have biological experiment. We put our samples in the microscope, and then we would go and create a big image database, no? Where we usually have to pre-process a little bit the images to normalize the sizes, the intensities, something like that. And then we would use some predefined measurements and predefined features to extract from each image a feature vector, no? And then every image, in this case, it will be represented by a feature vector. So this is the scenario that I was describing before. Now we can jump, for example, if we know the original categories, into a supervised learning cycle, where we first do feature selection, because sometimes some of these measurements, some of these features are not very informative, or they are redundant, or actually they are correlated to each other. So we can get rid of them and then have shorter vectors, and that way use less memory and train faster. Then we can select a classifier from the machine learning literature. And then based on our partitioning, we train in the training set, we evaluate in the test set. Then we check the result in test, always in test, okay? And let's say that we are happy with the result. What does it mean? We are happy because the accuracy is high, for example, and it is comparable with either the state of the art or human performance. Well, then it's great, because then we can go back to the real world, put more images in the microscope, go through the same process, extract the same features, and then apply the classifier without training. So that's the easy part. But most of the time, we are not so happy with the results, so we have to keep going into this cycle, revisiting the different steps, trying to improve the final result. So regarding algorithms, there are plenty. I told you this started in the 60s, so we have plenty of linear and non-linear solutions where they all try to do the same, find the boundaries in a more or less sophisticated way between our classes in feature space. Before deep learning, the most popular ones at the moment were support vector machines and random forest. But now, because deep learning is based on artificial neural networks, we'll go through a little bit the basics of artificial neural networks. So again, we have to a little bit jump back in time, actually to 1956, which is the very first attempt of an artificial neuron, which is called the Perceptron by Rosenblatt, which is actually a simple linear classifier. Okay, we're trying to find a line to separate our data points in feature space. The idea is very simple. We get a set of a matrix of weights that we multiply by our feature vector plus a bias. So we are defining the position and the orientation of that line. And if that result is positive, then we say it's class 1, for example, class A. And if it is not, then it belongs to the other class. This is a binary classifier. So it's very simple. You could say, okay, so why so much excitement about it? Well, actually, because there was, for the first time, an algorithm to train this type of approaches in an iterative way. And it's a very simple way. We take at initially random weights for that matrix. So we take one sample, apply the formula, multiply by the weights plus the bias, and if it is correctly classified, then we don't do anything because the weights are okay. If it is incorrectly classified, then we have to correct the weights, and we do it by the difference between the expected output and the real output. So iteratively, by feeding the algorithm with more and more samples, it goes correcting the orientation and the position of that line. Actually, it can only converge if the data is linearly separable, and it converges when it finds the linear separation. So this so-called perceptron, okay, it was exciting, but it's a linear machine, and many problems are non-linearly separable. For example, here I put you what we call in computer science XOR predicate. So we need two lines to separate the data samples here, right? So with one perceptron, we cannot solve it. So, okay, they found a solution, said, okay, instead of having just one single of these perceptrons, we can add what we call one layer. We're going to stack two more of those in between. So the first one calculates one line, the second one the other line, and the third one in the final layer combines them. Okay, so we can solve with one hidden layer, we can solve that type of problems. And actually, it was mathematically proved that if you include actually another layer, yet another layer here, with two hidden layers, you could, in principle, solve any classification and regression problem. So this is fantastic, right? Problem solved. Well, there was a little problem, and it was that at the time there were no algorithms to train this type of approaches based on something like the iterative samples that we saw before. For one perceptron, it was clear how to do it. With more than one perceptron, more layers, they didn't know how to do it. So this led to what it is known as the first neural network winter in 1969. And basically, artificial intelligence moved towards other kind of systems, symbolic systems, etc., who later led to the artificial intelligence winter in general. So we can summarize the early work on perceptrons by saying that the architecture was right, but nobody knew how to train them. This changed in 1986 with the appearance of this book, Parallel Distributed Processing, Explorations in the Microstructure of Cognition, Psychological and Biological Models, right? By a group of people in which Jeff Hinton was already there. Jeff Hinton is one of the current stars in deep learning. And behind this grand title, the very important thing that they did is that they transform learning into an error minimization problem. They move artificial intelligence towards optimization. So this was great because then we could adapt the weights of the multi-layer perceptrons by simply reducing the error on a specific function. Okay, so suddenly the multi-layer perceptrons became what they are, flexible and efficient nonlinear solutions for either classification or regression problems. How do they look? Well, like I put you here, they usually have one input layer. Imagine that you would have the feature vector in here, one value for each of those circles that are called neurons. One output layer, where we have the expected output or the probabilities of each class, or if it is regression, the specific numbers. And in between, one or more hidden layers. So how do we calculate this? For every of these neurons, for all the arrows that are right here, we get one weight. And to calculate the value here, we have to multiply the value from the input by its corresponding weight, okay, plus a bias. And then they usually apply an activation function, okay? It's also biologically inspired, and it allows us to actually also control the range of values that are output by one of these neurons. Okay, so this is all what we call a feed-forward process. We start from the input layer and go adding up until the output layer. So basically, we are defining a very highly nonlinear, weight-dependent, because everything depends on the weights on all these connections, transformation. And we calculate those weights by minimizing an error function. For example, we are doing age estimation with the facial images that we mentioned before. Well, we could take the minimum square error between the expected age and the actual age, for example. And how do we go from the error to the change in the weights? For that, we have a very elegant algorithm that is called backpropagation, because it backpropagates based on the gradient of the error, the error from the output layers towards the input layers. And using a chain rule, then you can adapt, or actually you calculate the contribution to the error of each of those weights. So you can adapt its weights accordingly. So this went on very strongly in the 90s until suddenly, well, new relevant contributions decreased. There were strong competitors, like I mentioned, the support vector machine or the random forest. And then this led to what is known as the second winter of neural networks. Actually, there was another big problem. That is that to attack significant problems, you need to use more parameters, to use more layers. And when you have 3 or more hidden layers, this is what we consider a deep network. We have the problem of the vanishing gradient. So when we were backpropagating the gradient from the output towards the input, the errors became more and more smaller. And at some point, like it was discovered, well, in the 5th layer, you have basically gradient 0. So stacking more layers wouldn't help because you couldn't adapt the weights accordingly. So everything changed in 2007 when 2 groups, one led by Hinton, one led by Benjo, using 2 unsupervised techniques, they show how to pre-train the networks, and then later, using regular backpropagation, how to fine-tune those networks. So suddenly the floodgates open, people started to use very large networks with millions of weights and parameters, new layers, initialization, regularizations, etc. So there was a completely new mood in the field, especially because, well, on the one hand, things that were impossible before were now much easier, and also they were leading to significant breakthroughs in many different problems, especially in computer vision and natural language processing, for example. 2012 is what most people mark as the starting point of the deep learning revolution, because the group from Hinton, they won the ImageNet challenge, which is this annual image classification challenge, with the use of a convolutional neural network called AlexNet, that actually won by a large margin. 2013, Google hired Jeff Hinton. 2014, Facebook hired Jan LeCun. That was the father of the first convolutional network. And this trio, Hinton, LeCun, and Benjo, they won the Turing Award a couple of years ago, which is equivalent to the Nobel Prize in computer science. So for you to understand deep networks, basically, I would recommend you to focus on two types of layers, right? One is the convolutional layer and one is the pooling layer. The convolutional layer is where we extract features from the images, which we do so by applying filters, by convolving filters with the images. In this case, I'm representing the convolution of a 3x3 filter. It's a 3x3 matrix with a small image. No? We just have to put it on top, calculate the multiplication of each value with the pixel values, we sum up, and then we get the new pixel values in the output image. So this is a way of extracting features from one image. If we want to do this at different scales, we have to somehow subsample the images. And to do so, we have the pooling layer. Okay, imagine that we have this image and we want to do a 2x2 pooling layer. So every 2x2 pixels, we select only one to go to the output. Which one? Well, if we select the maximum value, then it's a max pooling layer. If we select the average, it's the average pooling layer. Both have their applications. So when we talk about deep convolutional networks, what we are talking about is about combining, especially most of the time, these two types of layers. No, we have the input is an image, then we have a set of filters, no, a convolutional layer that allow us to extract features at that specific scale. Then we subsample. Then we have again convolution, so we get features at a different scale, subsample, convolutions, etc., until we get to a point where usually we get the information of the image really condensed, usually in a single vector, and there we can just plug in a regular multi-layer perceptron. And why is this useful? Well, because we can train this end-to-end with back propagation. We calculate the weights of the filters as well, and those filters are specific of the problem. Okay, it's not like before that we have to predefine some filters and then see if they were good enough and then use them in with a specific classifier. Here, everything is done for the specific problem, and usually with connections and weights in the millions. Is this useful? This is very useful, especially in computer vision applications where we can do this to obtain what we call semantic segmentation, classification and localization of single objects, multiple objects, everything at the same time, segmentations, detections, etc. And here I'm going to pass the word to the next speaker. Thank you for your attention. I will take questions, of course.
Sebastian.
Or if anybody wants to make a question.
Sorry, yeah. So I think all the questions were addressed in the chat. So in the interest of time, we will move Roma talk. So going to the practical part with zero cost DL for MIC, an open platform to simplify access and use of deep learning in microscopy. So Roma, I'll let you share your presentation.
Cool. Thank you very much. So let me just share this. I'd like to thank the organizers for having me today. I think it's a great meeting, and I'm really excited to talk right after Ignacio, especially because he's given us really an excellent overview of the field, and I can just piggyback on everything that we all know now. So what I'll be presenting to you today is our new tool, our new platform that we've developed for deep learning applied to microscopy. As you've seen from Ignacio just now, it's absolutely clear that deep learning has enormous strengths, enormous powers, potentials for bioimaging. But the bottom line is it's still difficult to use, and our tool, the tool that I'll be presenting to today, Zero Cost DL for MIC, intends to make this a lot more accessible, a lot more hassle-free. And so because Ignacio has already given you an amazing historical overview, the only thing that we'll add before showing you a few things is that the last couple of years, literally the last 24 months, have seen an enormous explosion of publications in the field of applying deep learning to bioimage analysis, to microscopy, to histopathology, to even natural sciences, right? But the issue is a lot of those work still come from computer science field, and they only slowly seeping into the field of bioimaging and biosciences in general. So I'll start by just answering this question. What is the fuss about? And first of all, it's useful to know what classical algorithms do. In the case of classical algorithms, you basically have an intention, which is in this case, say, denoising an image. You'll go from left to right. And what you'll spend time on is trying to design mathematical operations that will do what you want it to do, right? So that could be, say, a Gaussian filter on your image that will improve the signal-to-noise ratio slightly. In the case of deep learning, the concept actually turns all of this idea on its head, because it starts from the data, right? And then by first generating and obtaining data of what we want as input and what we want as equivalent output, right? We can feed this into a neural network and then actually tell the neural network, here you go, you're gonna have to learn how to go from input to B, from the output. And now you can work it out. And as we've just heard, there are plenty of algorithms, including backpropagation and so on, to allow for parameters to self-tune, to allow for this transformation to be learned by a neural network. Once the neural networks learn how to transform from those guys to those guys from the training data set that's provided here, then we obtain a trained model. And that trained model now can be used to actually process new data sets that will then perform the transform that we've trained it to do. So in this case, you see a clear denoising approach that's applied on similar type of data. So the fuss about deep learning is twofold in my view. One is that it can approximate actually any transforms, right? In the framework that you've seen today, there is no real limitations in terms of what you'll feed as input and output, as long as it can learn it, right? And we can discuss this afterwards if you want what this means. But that means that opens up for a lot of different tasks for deep learning to work, right? Even using similar architecture. A similar architecture could learn lots of different types of bioimage analysis tasks. And so that makes it extremely versatile. And so that's powerful from that point of view. But the second important aspect there is that neural networks also can massively outperform classical algorithms for a large range of different tasks. So you've heard about image classification, but also image denoising, image object detection, and so on and so forth. And that's why all of those algorithms are now used for self-driving cars, for image classifications on social media, and so on and so forth, because they are the most powerful ones. So that's good stuff, right? That's the good components about deep learning. The not so good component about deep learning is that training the models, so that step right here, which essentially determines how well it works and what it actually does, it requires two major things. One is still typically quite a high level of expertise in computational sciences, right? So it requires quite a lot of knowledge. And second, quite a lot of computational power. So typically, maybe a couple of GPUs, but at the very least, a number of servers to be able to run those within a reasonable amount of time. So and that's quite a difficult aspect to handle if you're going to be interested in running and using deep learning. And so these components right here actually can make people in the bioscience field, including us when we started working on this, really feel like training deep learning is a little bit like performing maintenance on the International Space Station. And what you see right here is Michael Hopkins performing actually maintenance on the ISS in this picture. So it can feel like this because it requires a lot of expertise, handle a lot of libraries, combination of libraries, installation, as well as handling hardware and software compatibility. And so what we intended to do with our work with zero cost deal for MIC is the following, is to really make streamline the capability of using lots of different neural network easily and really completely accessible to anyone from the biosciences. So let me give you a quick overview about the platform and how it works. It all starts with your own computer. I'm assuming you all have a computer because you're listening to me today. What you'll start with is your training data. That's always necessary, as we've just highlighted, but also your new data, the data that you want to process. And so our approach is to actually connect your local data with the cloud computing capabilities of a number of Google products, including Google Drive and Google Colab. And so the first step is to upload to the cloud all of your data. And then what we provide is a set of instructions taking the shape of Jupyter notebooks that can then be connected to Google Colab. And Google Colab is an online cloud computing platform that gives essentially for free a lot of computational power. So you have access to GPUs, local memories, and you can set up virtual machines all online. But the bottom line is by just hooking up the platform with the streamlined notebooks that we provide, then you can perform completely hassle-free network training because they'll be able to go fetch your data straight away from your Google Drive. You'll perform validation, extremely important step. Also, again, as Ignacio highlighted, a way to check the performance and the validity of the neural networks that are trained on the platform. And another step is then to perform the prediction. So take the new data and then obtain the results from the newly trained model out of it, right? And so the last step is to actually just download back all the trained network and the results, right? As simple as this. And you see that by shifting all the hard work, all the high computationally expensive steps of network training, validation, and prediction on the cloud, then there is no requirement essentially on what your machine is. So could you do it on your laptop and so on and so forth. But essentially what the platform provides is, first of all, a completely free cloud-based access to GPUs and computational power that you need to use deep learning for your microscopy studies. Second is a completely simple interface, and you'll see that in a second when we switch to the demo. And it requires literally no coding, but the code is absolutely accessible if you wanted to dig into how things work. And last but not least, it provides a single platform that will do all the complete workflow of deep learning, namely training, prediction, and quality control on the validity of the trained models in the platform. And that is quite important. And so from a user point of view, what this interface will look like is simply a web browser, and we'll take a look at that in a second. And so all that's required is a web browser. But what can the platform actually do? What we've done is only provide a platform, and we've integrated a number of deep learning networks that had been developed by the computer science, the bioscience communities in the past couple of years. Some of the most powerful methods out there developed recently, and then along a number of axes that can be really useful for bioimage analysis. One is to tackle the problem of image segmentation and object detection. So we've implemented classical UNet type of image segmentation. You can see here applied to electron microscopy data, both in 2D and in 3D. So you see here mitochondrial network being automatically segmented from a trained network where you see a good agreement between ground truth and prediction. We've also implemented Stardust, which is a neural network developed for the detection of nuclei from, say, microscopy images, including fluorescence microscopy, and that's extremely powerful as well. And another really quite exciting network that we've implemented is to be a YOLO V2, to be able to do object recognition, object detection in images. And so you see here from migration, bright field migration microscopy data set, we're able to then detect elongated, rounded, and dividing cells, and then follow them as the assay goes on. The second type of task that we've also implemented is image denoising and or restoration. And for this, we've implemented some of the most powerful denoising approaches, in particular the CARE networks that some of you might know. And you see here left the raw data. These are SIM data for which you see a little bit of patterning happening, and this was also gotten rid of thanks to that denoising and restoration approach that we've applied using the platform. Another type of approach is to use noise to void, also another network for denoising that's actually completely unsupervised, so that doesn't require paired data set. We've applied it to 3D live data set of mitochondria data in this case. And so you see here on the left the raw data, and the right the denoise data. And last but not least, we've also implemented a number of image-to-image translation, and that's something, in my view, that would be absolutely nearly impossible to do with classical algorithms. And in particular, one of the things that we've implemented is a network called FNet, or label-free prediction, that's capable of taking bright field images and predict the pseudofluorescence signal that you'd obtain from a fluorescence marker. And so in this case, we've got bright field images, and we've predicted the mitochondrial localization of those images of those cells from the bright field. So that alleviates somewhat completely the necessity to even have a fluorescent marker in there in some cases. That's extremely powerful. Another approach is to implement pix-to-pix. That's a GAN type of network. And in this case, we've actually demonstrated that pix-to-pix can be used to predict, say, the DAPI staining of say, a fluorescence image here, purely from the actin label, from an actin label image. And so what this means is that, again, you would be able, from the trained network, you'd be able to do away with having the DAPI staining altogether, and therefore extract nuclear marking or nuclear labeling straight away from just one single labeling. And so that's also another approach that, in my view, would be very difficult with classical algorithm. I'd like to wrap this up and thank everyone involved in the work. So that's obviously an extensive collaborative effort, including some of the original authors of all of those networks, and then basically the deep learning and bioimaging community. We've had a lot of beta testers also helping us setting all of those up. But without further ado, I'll switch to demo time. Okay, so demo time. Here we go. So what we've got here is our GitHub website, which is completely open source, and we'll share all the codes for all of those things. But what we've made this is as user-friendly as possible. And if you go on our GitHub page, you'll see a whole list of different networks here, and for which different tasks are possible, and the corresponding data set and so on. And I'll be demonstrating you how easy we've tried to make those guys by doing a live demo of those things. What we'll be doing today is do a simple segmentation of electron microscopy dataset using a UNet type of dataset. And so I'll open a notebook directly from the GitHub website. If Google Colab is going to then load this up for us. There you go. And so here's a quick overview of what the user interface will look like to any of you trying to use it. You've got the table contents on the left that will replicate the workflow of deep learning that you'll need. First of all, install all the dependencies, then set some parameters, train the network, do the evaluation of your model, and then using the trained model eventually. Okay, so let's. I'm going to take you through this. First of all, we're going to initialize the session by running that little bit of code. It's as simple as clicking on the arrow right here. And so what this first kind of bit of code will do is to, first of all, initialize the session with respect to Google Colab. And so that will also tell us a few information about what's going on with this virtual session. In particular, we've got access to a Tesla T4 GPU right here. The second thing is we're going to have to then allow this session to access data that we have on our Google Drive. So that's done right here. And so you see that we're simply giving permission for this session to access our data. And so I'm just giving it an access code. And just to show you, here's my my Google Drive. I've got a bunch of data in here for a number of different notebooks and about different networks. And I'll be using Unit 2D right here. And so the data that I've got for Unit 2D, sorry. So you see that now it's mounted. I can now click on the left, and what you see right here is I've got access to my complete drive, the complete Google Drive with all of my data on here. So I've uploaded already ahead of time training data and test data to play around with this for today. Next step is install the network. In the meantime, I will be showing you what I've got in here in my unit folder. Right here, I've got a bunch of training data, some images. I've got a folder where I'm going to store my models. So now we've got everything installed. That just took a few seconds. We're going to go through trying to set up a couple of parameters for training this network. And so the first thing is to give it the source images, so the initial electron microscopy images, and the ground truth masks that are paired with those. And so I've got two folders right here with the data in it. So let me just show you quickly. We've got here, I've got a bunch of images into my training images. I'm going to copy the path in here. And similarly, and with a one-to-one correspondence, I've got all the masks right here, and I can actually give it the path to the data. And so now we've given it all the data, so you'll know where to find it. I'm also I've got a folder for the models. I'm gonna copy the model with the folder for all the models right here. That's where I've chosen to save. That's where the trained model will therefore be saved. I'm gonna give it a name. Let's give it the name of today's date. I think we're the 26th. Let's say unit model 1. And then it's an iterative approach, right? It's going to go over and over those images, refining its parameters, tuning its hyperparameters to be able to learn eventually the transforms between electron microscopy and masks. This is what a number of epochs does. In the interest of time today, we'll actually only be setting up, doing it running for just a very few number of epochs, let's say 10 epochs, but a longer session would be really beneficial for the quality of the network. And there's a bunch of advanced parameters that we probably don't need to worry too much about today, but there are things, for those of you who have tested a little bit deep learning, there are parameters such as batch size, how many bits of images you'll feed at any one step of the iterative process, the size of the bits of the images, and also how much of your data you set aside for validation, right? And that's also important, again, in the light of the validation that Ignacio also mentioned to us earlier. So let's just set those parameters. Now by running this, the notebook's going to go fetch a couple of images, start looking through our folders, and then check that everything's in order. It should hopefully be in order. So now you see it's going through our data. Taking its time. There you go. And it's going to give us an example of what was in our folders. So that's an example of inputs and the equivalent outputs, right? And it's going to learn from that kind of data set. We've got the capability of doing data augmentation, which is a way to actually transform the input and output data in ways that are sensible for the data set that we are dealing with, and that increases the richness and the complexity of the data set. So I'll be using data augmentation today, but I'll be tweaking it a little bit. Let's get rid of some of this. And so all I'll be keeping in this case is you'll be flipping the images, so flipping 90 degrees and flipping by 180, and also flipping that by 180 degrees, and ignoring any other kind of transforms. And so you see how we can do this. It can kind of rotate things around and so on. And so that makes the training a little bit more robust. We've got another thing that we can set up to get started and to give the network a bit of a head start. To give it a head start, we can actually tell the network to not quite get initialized completely randomly, but start from a pre-trained network, right? And so we can do this by, I've already got a network that I've trained yesterday that I'm going to actually give it. And then what you'll do is you'll actually initialize all of its parameters to that pre-trained model so that the training starts from a reasonable starting point, and that'll help us get to a reasonable place today fairly quickly. But that's something that we've implemented in all our notebooks to be able to really take advantage of all networks that might have been trained by others in the field ahead of time. So now we've loaded this. We've got a little bit of initialization for this training to get started. So that step actually gives us a nice representation of the network architecture, and you'll recognize a few common type of layers, some convolutional layers and pooling layers, and sequence of those encoding and decoding typical of UNet. And then we can just start the training, and that's it. We are training a neural network from bioimage analysis, state-of-the-art neural network, online with 10 clicks, and completely for free, right? I've not paid anything to be where I am today, and because I've only set up a handful of epochs, it'll only be taking 1 or 2 minutes for this to happen. Are there any questions while we wait for this? Sorry, I don't see the question.
Yes, Romain, I will tell you. Yeah, so there is one question from Crisanta Preza. She's asking, how much can you trust the neural network result, especially in the case of the fluorescence inferred from wide field images? So could you comment on this, please?
Yeah. So I mean, it's essentially the question that everyone should be asking. So I'm really glad. The bottom line is, it's absolutely essential to perform quality control on any model. So the way to actually check whether you can trust your output is to make sure you generate some ground truth data that you can then compare to the prediction on a data set that the neural network will have never seen during training, and then check whether those the output matches the expected ground truth, right? And so this is one of the reasons why we've put a lot of effort into making sure that there's a step of model evaluation in this that does exactly this for every single network. So basically, the bottom line is, with a test data set for which you have the ground truth, you can quantitatively assess the quality if the answer is no, the quality isn't good enough, then you need to go back to training step and then maybe fine tune some of the parameters and then iterate again a few times until you get to a performance that you can trust and where you know where it fails and where you know where it performs well. But the important line is this platform actually really allows us to estimate and optimize that step. So I hope that answers your question. So now we've got the
Romain, do you want to take a quick question first? From Martin Kenny is asking how many images are required for training a pix-to-pix network, more or less.
So pix-to-pix is a bit of a funny beast because it's a GAN, it's a generative adversarial network, and in our hands, it's required a few hundred images, right? But in the case of the example that I've shown you earlier, going from actin network to DAPI network, generating the ground truth data set isn't a big hassle because it's simply throwing some DAPI staining on an actin, on a LifeAct kind of LifeAct expressing cells, and then acquire a bunch of two-color images.
Okay. And then John Halpin is asking if you have any plans to make an offline version that would run on your own GPU, because your Python notebooks are quite convenient to use. So yeah, he'd be interested in knowing that.
So offline without or local with local GPU. That's what you're saying, right?
Local GPU.
Yeah. So there's already basically a pipeline in place for running the notebooks on local machines. So there's local runtime capabilities, and for this actually there's we have a couple of extra steps that you can follow from our notebooks to here. If you want to run locally, Google Colab will help you. If this will actually, there you go. And there's a few explanations about how to do this. And in particular, if you're familiar with how to do this, the requirement, the text is automatically generated right here. So you can actually hook this directly to your local machine and get the local requirements right before you start on the local machine. So that'll allow you to make sure you have all the requirements in place.
Okay.
So we've streamlined this as well.
Thanks, Romain. I will just ask you to try to be fast on the last part because we are running quite late, but please go ahead. Yep.
Yeah, will do. I'll just show you just very quick things. One thing is we've just generated the model right here. That's what we've just computed. And one thing that we have also implemented is the following, that I've just downloaded this from, there you go, and that automatically generates a report from the training that we've just done, right, with all the parameters and so on and so forth. Okay, but now going back to this, I'll just wrap this up very quickly by just showing you two things. Is one, that we can evaluate the model, and in particular check the loss curves. So that's how the errors between the prediction and the test and the validation data set have evolved over time. Because we've only done 10 epochs, that's gonna look a little bit ugly. But importantly, we also have test images that we can also use for prediction, for quality control, as we've mentioned just earlier. And if I run this, I've got a couple of examples that were not included in the training data, but are similar, the kind of thing that I would be using for my predictions, the kind of data that I'll be using interested in using. And so for this, I also provide a mask. And for this, in this case, we now have the input data right here, the ground truth, the prediction data. So that's what we got out of the network, and therefore the overlay between the two right here. And so you see where it got it wrong, right? Where it's purple right here, it got it wrong. It missed out a few bits. And so this allows us to quantitatively assess the quality of the output. And we also have quantitative metrics to be able to do this. And with this, I'll just show you just 30-second, just the last thing. You can now use the trained data set, use the trained model right here by inputting some of your data and therefore exporting the new data that you're interested in segmenting from this model directly onto your Google Drive. But if you're interested in, if you're not interested in running this onto Google Drive, we've also recently worked closely with Estibalis, who's the next speaker, who's amazingly helped us set up a direct export of the model that we've just created. So right here, running those 2 extra bits. And so this export right here actually creates a model, or a model file, I should say, that is right here, that will also be saved into the folder that we've created for this model. And this new model right here can then be exported and fed directly into DeepImageJ for running inference and predictions directly on Fiji and ImageJ. So you could run easily training onto Google Colab in 0 cost, and then go run as many inference and predictions as you wanted onto Fiji. And with this, that's my transition to Esti. And thanks for listening.
Thanks a lot, Romain, for this very exciting presentation and also for the smooth transition to the next talk. So DeepImageJ, Estibalis, the floor is yours.
Okay, so oops, now. Okay, so thank you very much, Romain, for the introduction. Actually, it's great. And thank you also for the organizers, because, well, I'm kind of happy to show you today DeepImageJ. I'll tell you later why. So, as said, I'm Estíbaliz Gómez de Mariscal. I work at Universidad Carlos III de Madrid. And DeepImageJ, it's a project in collaboration with EPFL, with Daniel Sash, who is here today also with us, and also Carlos García López de Haro and Arrate Muñoz Barrutia. So as we don't have too much time, let's go. So as you can see from what Ignacio and Romain presented to you, is that there's a big challenge for deep learning solutions and for the people who are developing this kind of image analysis, and it is the path to integrate these pipelines into larger research workflows. So for that, we need we need user-friendly tools and open source tools. And actually, we kind of did this workshop 2 years ago, and it was not possible to show all these nice solutions that today we are showing to you. So that's why I'm happy to be here today. And we have some common software such as Elastic or Cell Profiler or 3D Slicer. We have integrated some deep learning solutions to keep working on their softwares with these tools, but also zero-cost deep learning for microscopy, as Romain showed to you, StarD, CSV ImageJ. So we can say that there's now something growing, and there's something working, so you can use your models quite easily. And for us, the target is just to run deep learning models, right, in ImageJ. And what we need is first to be functional. So we want to integrate new models, new trained models, and we want to process new data. But for that, we also need to be general. So we would like to be compatible with all different convolutional neural network architectures. So you can choose the task to perform, and you are free, to use deep learning without too much effort. So that's how DeepImageJ grew a year and a half ago or something like that. And what it consists is just an easy-to-use tool in ImageJ. It works like any other plugin. It is macro recordable, and it unifies the interface for TensorFlow and PyTorch models. So In green, I'm showing to you what we have in this new release that I'm not showing to you today because probably we will have many problems, but if it is not tomorrow, it will be next week in our webpage. So be ready for the updates. The second thing that is nice about DeepImageJ is that it's really out of the box. It's just one-click installation to get it, as any other plugin, but also for the models. It runs on the laptop, on a CPU, or on a GPU. There's also a GPU connection available for TensorFlow. And when we started this, it was kind of a proof of concept because we wanted to build a model repository, right? So as a developer, I train a nice model to perform a specific task, such as, I don't know, segmentation or some denoising. And I can upload my model so you can try it, and then we can, I don't know, train it again, or I can share my models with my collaborators and through the online page. And the nice thing is that this was a proof of concept, and now we have Bioimage models too that I will tell you about it later on. That seems to go on, and then it's going to happen, and it kind of works. And the last thing is that with DeepImageJ, as a developer, you can make your models accessible. And this means that they're easy to use, they are ready to use, and even if you think that it's difficult to create the bundle model as in the Python code, we have also created a guided user interface to build it. So it's not, it's like 2 minutes and you can provide your model together with your publication or to your collaborators without spending too much time on working on a user interface or anything else. So how does it work? We have a model developer who trains the model and creates a bundle model, just a file, as Romain showed to you. And this model can be shared into the cloud. So this is how the model repository look like in our web page. We have the models, you just need to click on download. Some of them, they provide Jupyter notebooks, so you can train them, or the code, so you can reproduce it. And the user just needs to install DeepImageJ as any other plugin, or you can download it through the update site, or install it using our webpage tutorial to do it, and download the model and just run it. And what does it have the model inside? Is just a folder with the trained weights of the network, the architecture, an example image, so you can have a demo. You don't need to provide the images at first, and the pre-processing and the post-processing written by the developer, who will do the work you need to prepare the images as inputs, or to just do some processing that you need afterwards with the raw output of the network. So for the user just needs to unzip this file, move the file to the folder, to the ImageJ models folder, and process it with DeepImageJ. Inside, what will happen is this. So you just open the image, and through the ImageJ macro, the image is getting pre-processed, so normalized, for example, here. And the plugin is going to do the prediction. So this is kind of transparent for the user. Finally, when we have the raw output, it could be sometimes, or most of the times, to do some kind of post-processing to get really the interesting part of the output that you need for your research. So there's also a chance to have a macro for post-processing and get the final here instant segmentation. And as you can see, all this, it's really a zero-code solution for the users. And the only part that might be interesting for them is the ImageJ macros that, that they are ImageJ macros. So it's kind of, if you are used to work with ImageJ, they are not very complicated to follow. But even though those are also given within the model. So now let's do a quick demo. Okay, here I have my Fiji application. And so first maybe I can show to you. This is the notebook executed that Romain showed to you, but with some more iterations. Here I have the output files, and this one could be the zip file that I need to download. So once I download it, I have it here, so I can just unzip it. It is somewhere here. And this is my Fiji folder. So I have my models folder here. If you don't have, you can just create it. And I can place this one just there. Now I can open Fiji. It shouldn't take too much time. Okay, here we go. So I'm going to use the example image given within the model. I have it here. And I can go to plugins, DeepImageJ, DeepImageJ Run. Here, I choose the model that I want to use. That is the one I just downloaded. I choose to apply the pre-processing given by the developer, the post-processing, and I click OK. And it just takes a few seconds. And I have this one. Maybe I should just do something like that. The output. So it's kind of easy to use. And also you can repeat the process. Just click on, let's say, for macro record. So I open the image again. And I can run it, choose my model, pre-processing, post-processing, click OK. And then I just need to copy these lines in a new macro so you can apply DeepImageJ to a batch of images in a folder or integrate it into a more complicated workflow. OK, so I'm just going through the presentation. OK, so where is the key for all this to work? It is in a configuration file, and this horrible text file contains all the technical details that we need to run the model in DeepImageJ. So this kind of stores all the information that the developer has to run the model in their Python code. And the reason why I'm showing this to you is because this is going to change. So what happened when we started working on DeepImageJ is that after just some time, we see that there was really a huge effort from the whole community to democratize deep learning. So to just show how should we use these models and how we should speak about them or interpret them, or how to do the validation or all these kind of things. You can see here all the open softwares that are really involved in this democratization. And actually, last year, there was a hackathon in Dresden organized by Florian Jack. And what they worked quite hard on was about how to build the bridges between Java, C++, Python, and JavaScript, you know, that are the languages that connect the user-friendly tools with the hard code of developers to train a model. And after a year, now we have the Bioimage model tool that I told you about. And one of the key components of all this to work is the configuration file that is going to be called the YAML file. So here you can see that are elastic, zero cost, CSV, ImageJ, and there's a huge effort being done in the last year to write all of us the same configuration file, so you can kind of use the models in different interfaces and we all interpret them in the same way. Also, the idea for this web page is that there's going to be a runner. Here is the runner for DeepImageJ, so you can try your models online to decide whether you want to download or not, or use it or not. So it's now the time for us to call you for some contribution. So if you have trained models that you want to share with, that you want to expose, or you just want to know how to put them on DeepImageJ or in the Bioimage model, so we will ask you to contact us or to contact through the forum or through GitHub, or I mean, there are many channels you can use. And I would really like to make this call for all of you who are listening, because I think this is, it's going to be important to share all these kind of deep learning tools. And here I just let you some links for further information if you want to go through deeper tutorials and so on. And of course, our webpage that is updated almost all the time. And special thanks for Carlos, Daniel, and Arrate, who are doing a great work to just develop and make DeepImageJ, you know, like general enough, and as I told you, functional. And all the people who has been really giving us recommendations, has many of the people here have contributed by very constructive reviews. So I would really like to say a special thanks. And that's all, I guess.
Thanks a lot, Esti, and also thanks to all the speakers for this amazing session. So it triggered a lot of interest. It's actually difficult to keep track of all the questions. So I don't think there are any unanswered DeepImageJ specific question. Correct me if I'm wrong, Daniel. I have one question for you, Esti. If you could comment on the future developments in DeepImageJ. For instance, I would like to see 3D processing for 3D nets, or actually 2D nets, or all these kind of good features, you know, and also GPU support. So I think it's already here, but maybe you want to comment on this.
Yeah, sure. So the current version of DeepImageJ is image to image. It is two-dimensional, but the new one will not have these limits. So actually, it works on classification, so image to result, 3D, channels, PyTorch, TensorFlow, GPU, CPU. So yeah, I can show you a classification example if you want. I don't know if we have time.
I trust you.
Eh?
I trust you. I will check it out.
You trust? Okay.
So Daniel, could you spot any question? So one question from Martin: does DeepImageJ use the integrated GPU of a laptop for generating prediction, or will it only work with Cuda GPU? Yeah, it only works with Cuda GPU, no? I'd say.
Yes.
Yeah. Maybe let's give 5 minutes to Esti to end and share the links when she's done. Yeah.
Okay. Maybe I can share the.
I think anyway, all the sessions will be recorded and shared somewhere. So, but yeah.
Okay.
But yeah, if you want to copy paste some links, I mean, about the presentation, I don't know how they will be shared or who will share them.
I will copy paste the link to the web page, and I'll put the presentation there.
Okay, a link to the presentation. Yes, if you're fine with this. Same for you, Romain. I don't know if you already.
I can happily do that, yeah.
And so, Daniel, do we have some unaddressed questions, or? I cannot hear you. Maybe wave your hand or in the chat.
Sorry, that's okay. I think.
Okay. Yeah.
Okay. Sorry. That's okay. There is no any more question. I think that we answer in the chat for the yeah.
Okay. So thanks again to all of you. And yeah.
I have a question for Romain. What about the train models from zero cost? Will they be available?
We've taken the approach of not really providing many pre-trained models, because really our intention before anything is the networks aren't ours. In some cases, the data isn't even ours. So we really want to get people to actually use it for training, right? And to actually take the time to download and try on the test data set that we provide by themselves, right? Most of them take only a couple of hours to do this, more as maybe an educational tool. For a number of specific models, though. We will be soon providing models, pre-trained models, notably for the big 3D stuff, like for 3D UNet, or maybe for or maybe for Stardust 3D and those ones that are a little bit trickier to set up. But apart from that, for the models that are simple to train, we probably won't. For the big 3D ones, yes. It'll happen shortly. And it'll probably go on bioimage.io just out of convenience as well.
One thing you didn't explain is how to contribute to zero cost, I think.
Yeah, that's a very good point. Yeah, I forgot to mention, we absolutely welcome anyone who wants to contribute notebooks. I'm sure a lot of people work on their own notebooks here and there. We have a page on our GitHub page that describes how to kind of use a template and then fit your model into our template, and then we integrate it as contributed notebooks. And absolutely, we welcome everyone to do this. There's only that many hands that we have on our side, and despite the fact that there are so many interesting networks that we could implement. So of course, please contribute. Maybe the easiest way is GitHub or email or Twitter. Thanks for the question, Ignacio.
No problem. I would say from my part, I'm willing to accept annotated data sets and not only networks. Or maybe people from the audience they don't have the notebook or the experience, but they do have the annotated data. And this is almost as important, I would say. Sebastian, we don't hear you. The micro.
Oh,
good point. Yeah, I was saying about the Google Colab free access. Do you know if Google plans to keep this free? Because I'm not sure it will scale up, like if many people start using it. So it's a good way to collect back unpaid tax money from Google as of today. But will it scale up and sustain? Do you know anything about this?
So, yeah, we don't have more info. We don't have any insider knowledge, right? And in fact, we've never been in touch with anyone from Google about this, right? Just to be clear. What we know is Google is rolling their Pro version in an increasing number of countries. It's available in the US and Canada now since recently. Yeah. So that seems to tell us that they are kind of making this a bit of a sustained product. We don't know whether the free version, of course, will remain free for very long or anything. But that's the way for them to get people hooked and then to move to the Pro version, which costs I can't remember. I think $25 a month to get twice the
it's quite
resources.
Cheap.
So it sounds like it's a tool that's here to stay, at least for the foreseeable future.
Okay, good. So let's close the session this time. Thanks again, and see you tomorrow. Bye.
Thanks, everyone.
Thank you very much.
Thank you.
It was a pleasure.
All take care.
Absolutely.
Yeah.
Good to see everyone.
Yes.
Stay safe.
Bye.
Good evening.
Be safe. Thank you.